Hello everyone, this is the last of the digestive system lectures, and we are going to talk about how the different macromolecules are digested, also known as hydrolyzed or broken down, and how they are absorbed. So digestion or hydrolysis means the breakdown of large molecules into smaller molecules. So mechanical digestion happens with the help of teeth in the oral cavity, which is cutting and tearing with your incisors, puncturing with your canines, and grinding by your molars. So mechanical digestion happens with the help of teeth, and chemical digestion happens with the help of enzymes, acids, and bile salts. So when we're talking about absorption, we are talking about small molecules, the monomers of larger macromolecules, which get moved out or absorbed from the digestive tract, so a digestive lumen, and into a blood capillary or blood vessel for distribution throughout the bodies. Now, some molecules are absorbed by simple diffusion, and others are transported across the intestinal wall using transport proteins. So we're going to start off by looking at the three groups of macromolecules. So foods that we ingest often have a percentage of all three of these. There are some food products like red meats or lean meats that don't have carbohydrates. You have certain foods that are carb-free. There are carbohydrate-rich foods. There are lipid-rich foods. So the three macromolecules we're going to talk about is first, carbohydrates. So carbohydrates are basically repeated units of C6H12O6, which is the monomer of carbohydrate, and that is called a monosaccharide. In terms of lipids or fats, lipids are made up of a glycerol backbone, and this backbone can either have 1, 2, or 3 fatty acid chains attached to it. So what you're seeing here is a triglyceride. So lipases, which are the enzymes that break down lipids, can break lipids down into fatty acids and monoglycerides. Proteins are complex structures, and the monomers of proteins are amino acids. So let's start by first looking at carbohydrates. So carbohydrates start with monomers called monosaccharides. A 2-monomer chain is called a disaccharide, and anything more than 2 is called a polysaccharide. And you have several enzymes, including one in the oral cavity that starts to digest starch, which is a polysaccharide. So the enzyme that starts off carb digestion in the oral cavity is basically salivary amylase. And this salivary amylase gets inactivated in the stomach due to the presence of hydrochloric acid. So stomach is a highly acidic environment, and so starch digestion that starts off with salivary amylase in the oral cavity is inactivated in the stomach by hydrochloric acid. So as the chyme is going into the small intestine, the duodenum, there are brush border enzymes and disaccharides that basically break, or disaccharidases that break down disaccharides and polysaccharides into monomers, which over here are the glucose, galactose, and fructose. Now, once they're broken down to these monomers, they're absorbed into the bloodstream. And the diagram on the next slide is where I'm going to explain how this happens. So what you're seeing here is an individual microvilliated simple columnar cell that lines the lumen of the small intestine. So this is where the lumen is. Here you're seeing your microvilli. This is the interstitial space. You have your blood capillary here, which is right here in the villus. And right next to that is your lacteal, which is a microscopic lymphatic tubule. So here you have a monomer, glucose, which is currently in the intestinal lumen. And glucose is actually transported, is co-transported, which is symported along with sodium here. So there's a greater concentration of sodium in the lumen, there's a lesser concentration of sodium in the cell. So sodium basically diffuses from an area of high concentration, which is here, to an area of low concentration, which is inside the cell. So as sodium is diffused from outside of the cell to inside of the cell, as is glucose. So the monosaccharides glucose and galactose are both transported or co-transported along with sodium. So right here you see glucose that is inside of the epithelial cell, and it is absorbed here into the blood capillary using facilitated diffusion. So it's co-transported with sodium from the intestinal lumen into the epithelial cell, and then it is transported by facilitated diffusion from the epithelial cell into the blood capillary, which is where it is going to go to the liver for processing and storage. So the answers to the blanks here, the monosaccharide glucose and galactose, are taken up into the epithelial cells and symported using a sodium gradient. So sodium diffuses, or diffusion of sodium down its concentration gradient, is what is causing that drag or pull for glucose to enter the epithelial cell as well. The next macromolecule we're going to look at are lipids, which include triglycerides, as we saw in that slide a couple of slides ago, as well as certain fat-soluble vitamins and steroids. So the enzyme group that digests lipids is basically lipases. So lipases are enzymes that digest lipid molecules and break them down into fatty acids and monoglycerides. And throughout our digestive system unit, we've seen three types of lipases. First is lingual lipase, that is secreted in the oral cavity. Next is gastric lipase, which is secreted in the stomach. And then you have the pancreatic lipase, which is secreted into the duodenum by the pancreas. Now, bile plays an important role in lipid digestion because you need bile salts for emulsification of fats. To emulsify fat is to turn large lipid droplets and chop them into smaller droplets. And this is because the lipases can coat more lipid, and the smaller droplets means more surface area for the lipases to attach and hydrolyze lipids. So I'm going to explain these two terms called micelles and chylomicrons right here in the next slide, which is where the diagram is. So here again is your intestinal epithelial cell. This is the lumen of the small intestine, interstitial space, followed by blood capillary and lacteal, which is all housed right here in the villus of the small intestine. So first, you see this yellow droplet right here in the center, which is your fatty acid and your monoglyceride that the lipids have chopped up into monoglycerides. is coated by bile salts. So these spike-like structures that you see coating your lipid droplet here is bile salt. So a small lipid droplet that is coated by bile salts is called a micelle. So micelles are basically endocytosed into the epithelial cell. So there's simple diffusion going on. So the micelle makes contact with the epithelial cell membrane and it enters the epithelial membrane, which is where you have your lipid molecule right here. And next, it is coated with proteins. So once inside the epithelial cell, intracellular protein coating the lipid is called a chylomicron. The chylomicron is then exocytosed into, now remember, lipids are hydrophobic substances. So to exocytose lipids, so they're not repelled, that is why you have these protein coats. So and protein is it's kind of like the bilipid layer of your plasma membrane of the cell. So this is exocytosed, goes into the intracellular space, goes into the blood capillary, and is absorbed by the lacteal right here. So chylomicrons actually enter the lacteals and are then carried through the lymphatic system, which is how they end up going to where the adipose tissue is stored in the body and deposit right there in the adipose tissue. Now, there are several proteases that break protein down. The first protease we see is secreted by the chief cell in the stomach, which is the pepsinogen. So pepsinogen is actually a precursor to the active form of the enzyme, which is pepsin. So when pepsinogen is in an acidic environment, it converts to pepsin, and pepsin breaks larger proteins into smaller polypeptide chains. So it's not breaking them into individual amino acids yet. It's breaking them into smaller, so like the first level of protein folding, so into the smaller polypeptide chain. Next, protein digestion continues in the duodenum, which is where the pancreatic proteases attack it. So the pancreatic proteases include trypsin, chymotrypsin, and carboxypeptidase, which also break down larger proteins into smaller, so the primary level of proteins, which is small peptide chains. And finally, the brush border enzymes, the peptidases from the brush border enzymes, are breaking it down further from peptide chains into ditripeptides as well as individual amino acids. Now, the way that individual amino acids are absorbed is something we're going to go over right here in the next slide, right here. So here's an amino acid, and it is co-transported, very similar to actually glucose and galactose. It is co-transported using the sodium gradient. So again, sodium, it has a higher concentration here in the lumen, a lower concentration right here inside of the cell. So sodium goes by diffusion from the digestive lumen into the simple columnar cell. So amino acid is co-transported, symported into the epithelial cell along with that sodium diffusion. Once inside the cell, it is absorbed into the bloodstream using facilitated diffusion. So this diagram shows you it's a great overview of what is digested in which segment of the digestive system. So in the oral cavity, you have the enzyme salivary amylase, which is a carbohydrate enzyme that breaks down complex carbs, which is starch, into polysaccharides and disaccharides. In the stomach, you have pepsin, which comes from pepsinogen, that breaks proteins down into polypeptides, which is at the first level of protein folding. In your duodenum, which is where all of the pancreatic and bile secretions have been purged, you have pancreatic amylase that continues the polysaccharide digestion. You have bile and bile salts that are starting to digest and hydrolyze lipids. And you have peptidases, your brush border enzymes, that are continuing the protein digestion. In the small intestine, specifically the jejunum, is where you have your disaccharadases, which continue to break down carbohydrates further until they're broken down to glucose and galactose. You have bile salts and pancreatic lipase that is continuing the lipid digestion, and peptidases, which are continuing to break down polypeptides into their individual amino acids. So the last part of the digestive system involves absorption of water, because chyme has a lot of water content, and most of it is actually absorbed right into the small intestine. So typically we are ingesting about 2 liters of water. There's 1 liter of salivary secretion during any given day, and gastric secretions as well. So this diagram is essentially looking at the source of fluid that gets dumped into your gastrointestinal lumen versus what is reabsorbed into your bloodstream out of the gastrointestinal lumen. So basically, there's less than 1% of water that is actually lost through feces. And conditions like diarrhea can result in water loss and dehydration, which is treated using oral rehydration therapy, so if you're having a lot of diarrhea and losing water, just ingesting water will not help much, but what you want is water that contains salts and sugars, so it helps water get absorbed into your bloodstream and not just passed through your feces itself. So I'm going to end this here. What we have not covered here there are several things we're going to specifically talk about in lab when we meet, and we will review some of this digestive physiology and histology of the digestive system as well.